Okay. So I hope the first slide is displaying very well. So we'll proceed with the presentation. So the title of this presentation is Evaluation of Progesterone Function in the Endothelium. And I am a professor at UCLA and have held a very long interest and long-term interest in trying to understand how endothelial cells are regulated by hormones and by growth factors. And the reason why in particular hormones are interesting is because, oops, I am trying to move the slide again. Very well. Okay. So the reason why in relation to angiogenesis hormones are important is because the uterus, which is regulated by estrogen and progesterone, undergoes cycles of regeneration that are cyclic, repetitive, and they're clearly coordinated by estrogen and progesterone. The importance of these hormones has been very well clarified in studies with knockout models in which elimination of the progesterone receptor or estrogen receptor results in devastating effects to the endometrium, not only to the endometrial lining as well as the regeneration of the endometrium as a whole, but also the inability of the conceptus to be implanted in the absence of these hormones. So we reason, as well as many other investigators, that progesterone and estrogen, specifically in the endometrium, also perhaps in the ovary, will play an important role in the regeneration of blood vessels and either both stimulating proliferation in angiogenic activities as well as promoting the maturation and stabilization of blood vessels. So for this reason, several years ago, we became interested in this question, and at the time that we started working on this, very few investigators had actually studied the contribution of progesterone. There was a lot more interest in estrogen receptor. And just as means as a quick introduction, we should probably review that progesterone receptor is in fact a relatively small protein that functions as a dimer. There are two isoforms which are encoded by the same gene. The PR receptor B, which is displayed there in the slide, is slightly bigger by about 165 amino acids than progesterone receptor A. And A obviously is generated through a splice form of on the same gene. Now, interestingly enough, even though these two molecules appeared extremely similar, the knockout of progesterone receptor B versus progesterone receptor A display very distinct phenotypes, revealing that in fact they are not fully overlapping, and they are actually interacting with different molecules. And there's a vast amount of literature in this particular subject, particularly in relation to epithelial cells, memory epithelial cells in particular, as well as tumor cells. So now, in terms of expression of progesterone receptor in the endothelium, we found a number of years ago, this is a paper or an image from a paper that was published in 1999, and in fact there is a Western blot there on the left. We found that a number of endothelial cells, human endothelial cells, express progesterone receptor, and the northern blot on the top then shows the stroma as well as a positive control, dermal epithelial cells that do not show expression, human lung endothelial cells which also show no expression, human endothelial cells which show very high level of expression, then human aortic endothelial cells much reduced, and finally human coronary endothelial cells. which actually showed a significant level. So in humans, it's clear that, at least by Western blot, we do see significant expression, particularly in the endometrial endothelial cells. But also some coronary arteries appear to express progesterone receptor. When one performs immunohistochemistry, which is shown now in panels A and D, is actually von Willebrand's factor, looking at normal as well as atherosclerotic lesions. And panels A, B, and C are serial sections of normal, and D, E, and F are serial sections of atherosclerotic lesions. And the red color is actually progesterone receptor. As you can see, the expression is nuclear. And the interesting thing is that the nuclear expression is not really contiguous. That is, not all the endothelial cells are expressing it, but it's more like a salt and pepper pattern. And that is very clearly seen if you can compare panels E and F, where the arrows in red are identifying nuclei in DAPI in the panel below that cannot be then seen in the upper panel with progesterone receptor, and that is in turn twined with white arrows that show expression of progesterone receptor. So furthermore, we have done binding assays as well as luciferase activity reported assays to demonstrate that in fact we do have activity of the receptor in the endothelial cells. And again, this has been published over a decade ago. So we felt fairly convinced that progesterone receptor was expressed in the endothelium. Unfortunately, the antibodies for progesterone receptor have not been very good available in the literature, and several other studies have questioned whether or not progesterone receptor has actually been expressed in the endothelium. So we felt that we needed to take different approaches to clarify whether or not, in fact, PR, progesterone receptor, is expressed in the endothelium, and for that we actually developed a mouse model in which the LAG-Z is placed downstream the progesterone receptor promoter, and using beta-galactosidase assays, we evaluated a number of endometrial sections, and there are two images that you can see there displayed on the top. These are from mice that are virgin. They have never gone through pregnancy, but very clearly you can see that the veins are very nicely expressing beta-galactosidase, indicating activity of the progesterone receptor promoter. And the arteries, which are indicated in A, are not showing receptor activity in the endothelium, but they are showing some activity in the cells that are surrounding the endothelium, that is the smooth muscle cells. So from this study, and I'll show you some more data, it seemed very convincing that in the mouse endometrium, expression of progesterone receptor was confined to the endothelium of venules at the exclusion of endothelium of arteries, but it was also expressed by smooth muscle cells and of both veins and arteries, and obviously it's expressed in stroma and epithelial cells. Underneath there is an endometrial preparation from a human uterus, and this is now looking at PCAM staining in red, and then in green, which overlays yellow, is actually progesterone receptor in these endothelial cells. So I think that we felt that this is sufficient proof that in fact there is expression of progesterone receptor in this subpopulation of cells. So what I'm showing you now is just another assay, also LAG-Z, and now you can see a lower magnification of the mouse uterus with the epithelial cells now showing beta-galactosidase activity, and all of the stroma population that is positive, and a higher magnification of that panel shows, indicating with the arrow, very nice endothelial cell positivity in these capillaries. And in the lower panels now, the larger venules and arteries, where you see expression in the smooth muscle cells of the arteries and veins, as well as in the endothelium of the veins. So the question then becomes, what is progesterone receptor really doing in the endothelium? And as the knockout animals were generated, a very nice paper published in Biology of Reproduction by Bertomali's group demonstrated that the endometrium of progesterone receptor knockout mice seem to have a recurrent high level of inflammatory cells. So this was removed from the publication, and again, this is a very old publication, and I do promise that there is new data coming. But what this old publication very nicely demonstrated on the left panels is a series of sections from a uterus, cross-section of the mouse uterus, of a wild-type mouse, and on the right is the same, essentially, magnifications, but now of a progesterone receptor knockout mouse, PERCO mouse. The staining is to evaluate neutrophils, and I hope that you can appreciate that, in fact, on the wild-type mice, there is a fair amount of neutrophils, which upon treatment with estrogen are not necessarily affected. But on the very bottom panel on the left, see, if you treat these mice with progesterone, there is a significant decrease of this population in the endometrium. On the right, now you can see the equivalent section, but now from a PERCO mouse. And the treatment with progesterone obviously has no impact, and these inflammatory cells are retained in the endometrium. So now this became very interesting to us, and we asked the question as to whether the endothelium, specifically the progesterone receptor in the endothelium, played any role in this retention or recruitment of inflammatory cells. So for that, I'm just going to essentially tell you that the next series of data is work that we are now in submission, and essentially tells the story that progesterone receptor appears to regulate leukocyte trafficking in the uterus by regulating the expression of endothelial leukocyte adhesion molecules. And this is very important because essentially it offers the uterus a mean of regulating trafficking of inflammatory cells in a way that is completely independent of the systemic circulation. So by virtue of expressing progesterone receptor in the vasculature of the veins and capillaries of the uterus, one can actually selectively alter how much inflammatory cells are coming in and out, regardless of what is happening elsewhere in the body. This obviously holds a very high importance for issues related to pregnancy, as well as immune privilege and protection of the conceptus. So the experiment now, all these experiments are done using a mouse model that we developed a number of years ago. And this was done by a postdoctoral fellow at the time, Minako Hashimoto Partika. And Minako made a conditional flux allele for the progesterone receptor. So she essentially placed LOX-P sites flanking exon 1. And we call that animal Percy for progesterone receptor conditional excision. And we crossed this Percy animal with constitutive VE-coherent Cre recombinase transgenic animal by generating then the progeny that contains both the Percy LOX as well as the V-coherent Cre we should be able to excise progesterone receptor specifically from the subpopulation of endothelial cells that that expresses this receptor. So we performed this experiment successfully, and I'm really skipping a lot of data to get into what is the most important finding. And in fact, we found, and this is work of a very talented graduate student, Lauren Goder, and also in collaboration with a very talented young assistant professor, Josephine Enciso. And what they did is they essentially evaluated whether or not there was a difference in the level of inflammatory burden in the absence of progesterone receptor in the endothelial cells. And what they found was that in analogy to the completely knockout mouse, they were able to reproduce this high level of inflammatory cells. So there is an increase GR-positive recruitment as well as accumulation in the uterus of the mouse, specifically in the endometrium, and it can be very clearly seen by low magnification there in panel B in comparison to panel A, but you can also see it very clearly under higher magnification. If these uteri now are essentially evaluated through FACS analysis using GAR-positive and CD11B positive, one can see that in fact there is a drastic increase in these cells, and these cells are not necessarily fully neutrophils, because as you can see they don't have fully segmented lobulated nucleus. They appear to be immature neutrophils, also have CD11B, so kind of a marker of macrophages. So they're myeloid cells that seem to be very important in populating this uterus, and they are found normally in a wild-type animal, but in the absence of progesterone receptor in the endothelium, there's a significant increase in the number of these cells. And that increase can be visualized here. So both Lauren as well as Josephine did a number of animals, and as you can see, the control, 11 animals on the control, show that the average is about 10% or so of the cells in the uterus. But in the PERCO, so this is full knockout, the number increases to 40%, and there are some animals that actually have very, very high numbers, and the p-value is statistically significant, as you can see there. Now, when we remove the progesterone receptor from the endothelium alone, this number is lower than in the full knockout, not surprising. However, it is still statistically significantly different from control, and it reaches around 20%. So it's almost twice as much as, it is twice as much as the wild type. Now, interestingly, this is only specific to this subpopulation of cells. Is not really affecting other cell types like macrophages. So this is a staining for F4AE, looking at wild type and endothelial cell knockout. And as you can see, the relative number of cells is really not all that significantly distinct. If you now look at T-cell recruitment with anti-CD3, now what we see is the opposite picture. So what we have here is in the wild type high level of lymphocytes and T cells specifically, and in the echo that is not necessarily the case. So it seems that these absence of progesterone receptor is upsetting the balance of inflammatory cells influx and retention in a way that we are having high levels of neutrophils or immature neutrophils, GAR-positive, CD11 positive cells, and a decrease in T cells. So in order to actually sort this out a little bit more clearly, we also perform additional experiments, and here Josephine and Ciso in particular did infection of the uterus with chlamydia mordarum, and chlamydia infects the uterus very effectively. If we infect wild-type animals, you can see there is a huge increase of inflammatory cells. But when we infect the echo animals, this inflammation is overwhelming, and in fact, it can be so devastating that the entire uterus becomes occluded with massive amounts of inflammatory cells, and that is displayed in panel C, D, E, and F. So in fact, the cytokeratin staining is just to show that there is a lumen in this uterus, but there is a huge, massive amount of inflammatory cells that form abscess and can compromise the viability of the uterus. So this trafficking of inflammatory cells is really important for not only actually pregnancy, but also for clearance of inflammation. So here's where we use the Fluxion system, and it became quite obvious that it was very important for us to start understanding how progesterone might be regulating the levels of different levels of inflammatory cells. And I hope you can see here in DAPI the nuclei of endothelial cells. And now in the bottom is actually vehicle exposed endothelial cultures, and the dots in green illustrate inflammatory cells that are attaching to the monolayer of endothelium. And clearly, the control here, the positive control, will be treatment with LPS. In 8 hours, you can very clearly see that there is a very obvious difference between the absence of LPS and the excess of LPS. And now, if we do these experiments in the presence of progesterone, we can observe that there is, in fact, a response. So that is, so in the absence of LPS, presence or absence of progesterone does not really affect the number of CD45 positive cells that adhere to the endothelium. But if we actually now expose these monolayers to LPS, absence of progesterone shows very avid binding of inflammatory cells, which is significantly abrogated when these cells are treated with progesterone, indicating that in fact there is an anti-inflammatory effect that this hormone is mediating. I think that the preceding slide shows that better. What I want to show you is actually how the experiment is done here. And what we do in GFP, actually, the endothelial cells in green are showing the cells that are expressing the progesterone receptor, because we have transduced them with lentivirus expressing progesterone receptor and also GFP. And then the CD45 cells are in red and the DAPI in blue. So this is a low magnification. It's very difficult to see, but nonetheless, this is the actual data. And now you can see that at 4 hours, there is clearly in the plus LPS, there are more neutrophils and CD45 positive cells bound to the endothelial cells than when we treat these monolayers with progesterone also for 4 hours. So it doesn't really take that long to actually have an effect. And in fact, if we now try to understand what is the specific molecular underpinnings that lead to this effect, we found that when we expose endothelial cells with interleukin-1 beta, this is the same thing that is identical we'll be seeing with TNF exposure, and then follow that with vehicle or progesterone in a time course, you can see that PCAM is not necessarily altered. Interestingly enough, ICAM1 is not significantly altered, but VCAM is very clearly modulated, particularly at 8 hours and 16 hours when the cells are exposed to progesterone. So bottom line is that what progesterone really is doing is selectively affecting some inflammatory cells but not others. And in this case, you can see now the comparison between VCAM and ICAM, and this is messenger RNA. The preceding slide was protein, so obviously it was a little bit delayed. The messenger RNA is rather quick, and as quickly as 2 hours, there is significant reduction in VCAM when we treat with progesterone. Interestingly enough, that's not what happens with ICAM, and in fact, in some cases, we see increase of ICAM when we treat cells with progesterone. So we believe that this selective effect, and we're now expanding this studies to looking at a wider array of inflammatory cells in endothelial cell adhesion molecules, will probably play an important role at selectively capturing some inflammatory cells who are not capturing others, explaining why we have an excess of GR-positive, CD11b positive cells and a decrease of T cells in the uterus. So we're coming to the end. Essentially, I want to conclude, a progesterone receptor is expressed by endothelial cells and is restricted to specific vascular beds, that is in the veins but not the arteries. We also show that PR in the endothelium regulates the trafficking of these inflammatory cells, and the regulation of inflammatory cells relies on a specific regulation of specific cell-cell adhesion molecules by progesterone receptor. So finally, the most important slide obviously is actually the acknowledgement, so you can see the group, and more importantly, Lauren Goddard, who has been really the champion on this project, and I only showed a window of it, and Josephine Enciso, who has been helping in particularly the Clostridium meridianum was all her project, and Amy Tom, then she was an undergraduate, a graduate student now at NIH. So with that, I would like to conclude, and if we have any additional questions, I'll be very happy to answer.